Hello class, welcome back. In the last video, we had a technical difficulty, so I want to jump right back into where we left off, which is a Punnett square with dihybrid cross, or two different genes. So go back over what we talked about. In this example, we're looking at the gene for seed color, yellow or green, yellow being dominant, green being recessive, but we're also looking at wrinkled or round seeds. Round being dominant, so that's big R, wrinkled mean recessive, mean little r. And we're doing a dihybrid cross for both genes right now. Now, before we get into how to make this big square, remember that normally with the genotypes, let's say big R, little r, the two alleles are separated. So big R separated, little r separated. And it'll be the same with big Y, little y. Big Y separated, little y separated. But keep in mind with the law of independent assortment, all these alleles are going to be combined in different ways. But you'll always get one of the R's and one of the Y's. So the way I like to do this is I'll write big R, little r, and I do both twice. You're going to see this big R could get a big Y right here, or a little y. The little r can get a big R or a little y. And we're doing one of each letter, one R, one Y. And these are the four combinations that can be made in terms of the alleles that are in each gamete. So in the female, for the eggs, we can get big R, big Y, big R, little r, big Y, and little r, little y. The same thing for all the sperm, since it's also heterozygotes. And then when we use the Punnett square and we bring them down, we bring down these two, so we get big R, big Y, bring over these two, big R, big Y. And it's the same if we choose a different one. So if we choose, let's see, still same example, I'm gonna bring this all the way down to the bottom. We have a big R and big Y, and we bring this one over, little r, little y. So same idea as a Punnett square, but we have two letters per gamete. Now that's one way to do this. And keep in mind, this is again due to the law of independent assortments. The segregation of alleles of one gene does not influence the segregation of alleles of another gene. So if we go back, the R's do not influence the segregation, the separation of the Y's. So you're always going to get one R, one Y in each gamete. You'll get one version of the allele, one version of the gene in each gamete, since you have a diploid cell, two sets of copies of the DNA. Now, if you don't want to do that big square, another thing you can do is what's called a product rule. And this is actually what I prefer because I believe this makes things a little easier. So let me go back real quick. Go back to this slide and change my color. So with the product rule, it simplifies these problems very, very easily. So what it does is we are going to look at one gene at a time. So in this first example, we're looking at just R's. If we did a dihybrid or a cross with a heterozygous, we know that we have big R, little r, big R, little r. So we do that cross. Say we're looking at the heterozygous. So we know half of these offspring are heterozygous. And in the next one, we look at just the Ys. And say we have a pair, again, heterozygous. So we get big Y, little y, big Y, little y. And again, we get half of these that are heterozygous. So we get half the offspring can be heterozygous. And then we're adding another gene, which is T. And again, we're doing a heterozygote for the T's. So we get big T, little t, big T, little t. And we cross, we get two heterozygotes. And we get half of the offspring can be heterozygotes. So now with the product rule, one, you do the Punnett square for each gene. So I'm gonna write that down somewhere. Step one. Zoom in. Number one, Punnett square for each gene. And then step two, multiply the results. together. And I wanted to go over that over the next slide too. So let me go to our next slide. So let's see. And the example again, we're taking two heterozygotes parents for R, Y, and T, and we want to see how many of those offspring become heterozygotes themselves. So with that, we know that for the R's, half offspring are heterozygotes. For the Y's, it's same. For the T's, it's same. So once you do the Punnett square for each gene, get the results, you're going to multiply them together. So 1/2 times 1/2 times 1/2 is going to give us this 1/8. So this is one that I'm actually going to do with y'all because I want to explain how it's done. So we have a male genotype with this big Q, little q, big B, little b, big D, little d. Is crossed with a female with a genotype this little q, little q, little b, little b, little d, little d. It's a homozygous recessive across the board. What is the portion of the offspring will be homozygous recessive? So we're looking for homozygous recessive. So what we're going to do is do each gene separately. So first gene, big Q from the male times little q from the female. We do our Punnett square for the male, big Q, little q, little q, little q. So we drag everything over, we get a big Q, little q in this one, big Q, little q in this one, little q here, little q here. And if we want homozygous recessive, we have 2 out of 4. So 1/2 are little q, little q. We do the same for the B's. So the second cross, we have big B from the dad, little b from the mom. If we do the Punnett square, big B or little b for the mom, big B, little b for the dad. It's the same thing. With the first box, we get the big D from the big B from the dad, little b from the mom. Big D, little b, little b on the bottom, and little b on the bottom again. We get 2 that are homozygous recessive. Remember, that's what we're looking for across the board. So that's 1/2 our little b. And lastly, we got one more across because we have 3 genes here. And we're looking at the D's. So we get big D, little d from dad, little d from mom. One more Punnett square. We do big D, little d for dad, little d from mom. And we do 1 square at a time. We get the big D and little d here, big D, little d here, and the bottom, little d, little d. And again, 2 are homozygous recessive, so we get 1/2. All right, little d. Now we're going to take all of those numbers and times them together for the product rule. 1/2 times 1/2 times 1/2. And for this, if this is on a test or anytime, use a calculator. This will give you 1/8. And that's the probability that you're going to get a homozygous recessive offspring in this cross for all genes. Oh, I circled the wrong number. That was my fault. Make sure you are careful, unlike me, who went and circled the wrong number. All right. For the assignment this week, it will help you with crosses. Use that weekly assignment to get used to Punnett squares. How does the law of independent assortment reflect the event of meiosis? For this, I want you to think about it, but we're going to move on to. So now we have a couple more things before we finish up today. Genes close together on the same chromosomes are linked. So I'm going to highlight that. Genes close together on the same chromosome are linked. Punnett squares and the product rule assume 2 genes are sorting independently. They do not influence each other. When genes are linked, they're physically near each other. Therefore, they do not sort independently. So when genes are linked, they're physically near each other. They do not sort independently. Linked genes are not inherited independently from each other. So there are some genes that if we look on a chromosome, So here's my chromosome. And say we have, oh, come back. We have two genes. So say we have a gene here for big A and a gene here very close to a little b. Since these two genes are on the same chromosome, these are what's called a linked genes. They're very close together. That means whenever this chromosome is separated, it's unlikely that these two genes are going to be separated. So the closer the genes are on the chromosome together, the more likely they're going to be linked. So when we talked about independent assortment and what the gametes are going to get, these gametes are going to have big A and little b, most likely. So they're going to have a big A and little b paired up together. So genes on the same chromosomes are linked. Punnett squares and the product rule cannot be used if genes are linked because inheriting one allele influences the likelihood of inheriting the linked allele. So again, with here, inheriting this big A influences the chances that the offspring is going to get this little b as well because they're so close on the same chromosome. If you get the big A, since that little b is right next to it, it's a good chance you're going to get that little b. Now, if that little b, this gene right here, was on a different chromosome, that would be different. We could use independent assortment in the product rule. But since they're so close, we consider them linked, and thus we cannot. All right, so now we talked about the Punnett squares, we talked about dominant and recessive genes. Now let's mix it up a little bit. Oh, so not all genes follow Mendelian inheritance patterns. So far, we've discussed genes with two alleles, in which the dominant allele masks the recessive allele. But alleles do not interact this way in all genes, which leads to alternate patterns of inheritance. So here, we have a red flower and a white flower. When they are mixed, we get pink flowers. So it's neither red, the dominant, or white, the recessive. It's mixed. This is what's called. So with that, the interaction of alleles determines the pattern of inheritance. Dominant alleles mask recessive alleles. In genes, Mendel studied, creating patterns of phenotypic and genotypic ratios. Remember, phenotype is the physical characteristic, what it looks like. And genotype is the genes, what alleles the offspring has. In other genes, alleles interact differently. This changes the nature of phenotypes, but not genotypes. So when we're talking about the next couple of things, genotype is staying the same. It's just the phenotype is changing. The first thing that's going to change is or first example of this is incomplete dominance. Incomplete dominant alleles do not mask each other. In incomplete dominance, nope, let me get my pen now. The heterozygote has an intermediate phenotype. Here, the red allele does not mask for the white allele. If they did, the heterozygotes would be red. So here we have two pink flowers being red. We have the alleles coming down, and we can see here we get one red plant, one red flower, one white flower, but then we get these two pink that are kind of in the middle. So that tells us, hey, something's going on. The red and the white aren't dominant to each other, they're mixing. So there's incomplete dominance. There's not a complete one allele isn't dominant over the other. They mix. So in incomplete dominance, you get a mixing of the phenotypes. And I'll write that down as a note here. Results and the mixing of the phenotypes. So in the example, we're mixing red and white to get pink. So pink is the combination, that's the heterozygote. Incomplete dominant alleles create a blended heterozygote phenotype. Is kind of what I talked about in the previous slide. When the red allele R1 and the white allele R2 are both present, the heterozygotes R1, R2 phenotype is an intermediate pink. The white allele encodes for a non-functional pigment protein. So again, in this, the red allele R1 and the white allele R2 are not dominant to each other. So their phenotypes mix, creating incomplete dominance and creating an intermediate phenotype, a blended phenotype. And that's only in heterozygotes, though. So the next example is codominance. Codominance alleles do not mask each other. And codominance, let me get my blue out real quick. More than one allele encodes for a functional protein. So in codominance, all the dominant alleles are present. And a good example is blood type. So blood type, we have type A, type B, type AB, and type O. And the genotypes are here. So we can see here in our genotypes, for type A, we can have big I with a big A, big I, big A, big I, big A, or little I. And the second one in type B, same thing, but with B. Big I, big B, big I, big B, big I, big B, or big I, sorry, big I, big B, and little I. So we have a little I too. But in this example here, we have both big I and big B. So you may think, okay, which one's dominant? Well, neither are dominant to each other. It's like, okay, so it's incomplete dominance. Well, no, there's not a blending of the phenotype. Both A and B proteins are present. Since both phenotypes are present and there's not a mixing, this is codominance. Both phenotypes are present and they are not mixed. And I can write that down at the top here. Both phenotypes present with no mixing. And that's the big difference with this, with codominance and incomplete dominance. Incomplete dominance, you get a mixing, and codominance, you don't. So with this, we know big I, big A is a dominant allele because it's masking little I, recessive. But big I, big B is also dominant. So the little I. So these two dominant alleles, when mixed here, are both present. So this is codominance because A and B proteins are located on the cell. So dominant alleles create a double phenotype, and that's what I talked about. If two dominant alleles are present, both proteins encoded by these alleles will be represented in the phenotype. So in type AB, you have the allele for the protein A, and allele for protein B. So we get protein A and protein B on the cell. We don't get a mix. We get both in code and codominance. Human blood cell types, alleles A and B are codominant. In human blood types, both IA and IB are dominant. If the genotype is IA and IB, confers red blood cells that contain both A and B molecules. And that's how we talked about with this type AB blood. We get alleles for both A and B, both are dominant, but there's no mixing. So you get both A and B proteins on the cell. Human blood type alleles AB are dominant over O allele. The I gene will also has a recessive allele, little I, which encodes a non-functional protein. But the two dominant alleles, IA and IB, make gene I codominant. So it's saying that we still have that recessive, that little I. And the recessive in homozygous recessive results in type O, where we get no proteins. So both A and B are dominant over little I. One gene, many phenotypes. So this is another thing that can kind of throw off our classic Mendel genetics. In pleiotropy, one gene has multiple effects on the phenotype. For example, a gene might affect more than one biochemical pathway. So there are some genes that can affect multiple phenotypes at once. So that those genes are called pleiotropy. And this again will throw off classic genetics and classic inheritance because that one gene affects multiple things at once. Now, on the flip side of this, we have what's called epistasis. So the products of different genes can interact with each other. So with this, go back real quick and check something on my teams. Okay, here we go. With this, one gene can affect another gene. Epistasis occurs when the product of one gene affects the expression of another gene. Expression of the H allele gene 1 affects the expression of A, B alleles in gene 2, which is blood type. So remember we talked about blood type, which is these proteins on our red cells, the AB, which gives our blood type. But we have this other protein that codes for this little part right here. So with that part, we can get a dominant and recessive gene. The dominant, big H, gives us this little protein. And thus we can express our blood type off it. But say we have the recessive version. The recessive version gets rid of that protein. Without that protein, we no longer have a blood type because that protein for blood type has nowhere to bind to. And that's called epistasis, when the gene for H influences how the blood type is, if the blood type is produced or not. So in epistasis, one gene affects the expression of another gene. If the first gene is not expressed, then the other gene is not expressed by default. No matter what the other gene is, so it's just blood type, it cannot be affected. It cannot be expressed. How does pleiotropy, epistasis, incomplete dominance, and codominance increase the number of phenotypes? Think about it. We'll come back. Okay. So for this, all these affect how we look at um, inheritance. With inheritance, if it's classic Mendel genetics, we have dominant and recessive alleles. We don't have a mixing. We have two phenotypes. But in all these, we can get a blend of phenotypes. We can get both phenotypes. We can get one gene that affects another or one gene that has multiple effects. So all of these different examples give variation in phenotypes due to how the genes are being inherited and expressed. So sex-linked genes have unique inheritance patterns. In humans, females have two X chromosomes. In males have one X chromosome and one Y chromosome. So X, sex chromosomes equals female, XY chromosomes equals male. I do want you to know this because it can be important very soon. This Punnett square shows that each fertilization event has a 50% chance of producing a female and 50% chance of producing a male. So we're talking about this right here. So in this, remember, with these two genotypes, we are splitting the letters up. So in the female, we get X and X, and the male, we get X and Y. Remember that Y produces males. So in this bottom row, we get two males, and the top row, we get two females. So in with sex chromosomes, when they're passed down, it's automatic 50% male, 50% female. Keep in mind, males only have one X chromosome, though. That's going to be important soon. Which gamete determines the sex of the offspring? The egg will always carry the X chromosome. So the eggs, remember, females are XX. That means a female can only pass an X chromosome. But males are XY. So the male can pass either X or a Y. So it's the males that determine the sex of the offspring because they can pass down an X or Y, versus females can only pass down X. The sex chromosome in sperm therefore determines the offspring, if the offspring is female or male. Males only have one allele for genes on the X chromosome. Males only have one X chromosome. Whichever allele males have on their single X chromosome is the one that is expressed. So if we're looking at these examples, we're looking at the X chromosome, it either has a big R or a little r. You can see in the female, we can pass two different X's. So in this first row right here, these females can have two different alleles for R since they have two X chromosomes. But in the males, males are XY. So they only get one X, one version of that allele because of that. So females get two X's. So a X-linked gene, they get two variations of the gene. But in males, if for an X-linked gene, they only have one X chromosome. So they only get one allele for that gene if it's linked to an X chromosome. Females have two alleles for the genes of the X chromosome. Females have two X chromosomes. The interaction of the two alleles determines the phenotypes. Here, the R allele is dominant over the big R is dominant over little r. Just like I said earlier, females have two alleles. We can do the classic dominant and recessive idea. So big R is present. That phenotype is always present, always produced. Where in males, we only get one X. So you have one allele, big R or little r, depending on what the female has. X-linked recessive disorders affect more males than females. Males only need to inherit one X-linked recessive allele to express the recessive disorder. Females must receive alleles in both X chromosomes to express the recessive disorder, which is less likely to occur. So if we have a disease that is recessive and only is linked to the X chromosome, females here have two X's. So it's going to be harder for them to get two recessive alleles. However, in the males, since they only get one X chromosome, it's a lot easier for them to have a chance of getting the disease because they may only get that one recessive allele, and that's it. So males are more likely to be affected by X-linked recessive disorders because they only get one X chromosome. If they get their recessive allele, there's no other version of that allele. That's the only one. So they are more prone to get X-linked recessive disorders. Hemophilia, I'm going to go back real quick. One thing I want to point out is this. So we have a female here that's X, big H, X, little h. She's a heterozygous. She won't express the disorder since it's recessive and she has this one dominant allele. But she's what's called a carrier. A carrier is a person that has the gene for the disease, but it is not expressed. So this female does not have the disease because she has a dominant allele, but she's a carrier because she still has the recessive version. So carrier is someone who doesn't express the disease, but has the allele for it. Hemophilia is an X-linked recessive disorder. If an affected female has a disease and an unaffected male does not have the disease, have a boy, what are the chances they will have hemophilia? So hemophilia is recessive. The female is affected, so we get X little h, X. Well, actually, I'm gonna have y'all do this. Pause, think about it. We'll come back to it. Okay, so the female, like I said, has the disease, so she has X little h, X little h. The male does not. So the male has X big H, Y. So when we do that cross, X little h, X little h, X big H, Y. Now we want the boys. So we're only going to look, I want to cross these out. We want to look at the bottom because we're looking at this Y. We get X, little h from mom, and the Y. And another X, little h from mom, and the Y. So both offspring have the little h. That means 100% of the males are going to have hemophilia because that's all the mom can give you. The dad only gives the Y. There's no way that dad can give away the big H to the female, to the male offspring, excuse me. because they have to give the Y to produce a male. All right, last thing we're going to go over. It's called a pedigree. A pedigree is a graph that depicts the relationship of genotypes and phenotypes within families. And you're going to see it like this image below. So in this image, it looks like a branching tree, starting with the first generation, which is the parents, then the children, and lastly, grandchildren. And a couple of things to note here. Females are going to be circles, males are going to be squares. A horizontal line that connects to means they are married. And then when they go down, that means these are the offspring for that couple. And then with this key here, unaffected individuals for a disease are going to be blank, carriers will be a mix, half and half, and affected individuals are solid. So a pedigree is useful because then you use it to track whether a gene is dominant, recessive, X-linked, and so forth. So in this example, we have an unaffected female and an affected male. And their offspring, notice how we have no carriers. We just have three offspring that have the disease. So that should raise red flags in case something's going on. There's no carriers. The three offspring who have the disease or have the gene are affected no matter what. And both male and females are affected too. So, and then if we go down, they are also passing out the gene too, no carriers. So keep in mind, a recessive gene, a recessive trait, has to have both recessive alleles to be present. So in this case, since the original parents, one did not have the disease, one was unaffected, one was affected, but the offspring who were affected had both, it wouldn't make sense. Well, let me go back. So with this, it kind of shows that there's no carriers at all. Since there's no carriers at all, we can say that, make the assumption that if a offspring has the gene for this disease, it's automatically present. And what that means, if the gene is always present, if present, then it's going to be a dominant disease. Because say if the gene for heterozygotes wasn't present, we would get a half color. But we don't get that. So this is going to be a dominant gene. Pedigrees show modes of inheritance. So in our first example here, this pedigree tracks an autosomal recessive disease. What I want you to notice from right off the bat, in this we are dealing with autosomal dominant. And autosomal dominance, we do not get any carriers. If the gene is present, the person is infected or affected by it. And an autosomal recessive, we are getting carriers. So we do have carriers for the disease, and we have a lot more carriers than normal. So with this one, we have a carrier here and a carrier here. When they have offspring, we get a carrier and a carrier still, but no one's affected. But when these two carriers are affected, we get two offspring that can have the disease. So we have represent this by big A, little a, big A, little a is a carrier is going to be heterozygous. They'll have the normal gene and the affected gene. Normal gene is dominant, affected is recessive. And we know their partner, it has a big A at least. So that means we get big A something or little a for the two carriers. And since they're big A, little a, they do have a chance of both passing down that recessive gene to give the recessive disorder. Now, in the next pedigree, this pedigree tracks X-linked recessive disorders. So here we have the female, so we have big X A, big X little a. A carrier, again, is heterozygous. It has one good allele, one bad allele. And the male is big X, big A, Y. So in the females, they're always going to have an A. So that's why we have these two females unaffected and this one just a carrier, because it still has this allele. But in the male, we can get it. We can't get that X-linked recessive disorder because the mom either passes the X, the big A, or the little a. So in this one, we can see that the males are always having it. There's no carriers in males, but there's carriers in females. So this is a red flag. And the big difference with the first example is we have carriers who are males and females. Carriers who are males and females can suggest a recessive disorder. But carriers that are only females and males that always have a disease if the gene is present should be a red flag. It's like, hey, this may be X-linked because the males are affected more than the females. So let's go over this question, and I believe, oh, already went. So let's go over this question. This pedigree tracks an autosomal dominant disorder. What is the genotype of I2? So we're looking at the genotype of this one. Pause, think about it, we'll come back. This is a harder example. So with this, couple things to know. There are no carriers here. Absolutely no carriers. And we don't have everybody with the disease. We have 3 offspring that have the disease and 2 that do not. So with this, we should be flagged that, hey, and 1, we have 2 females that are affected too. So with this, we should be flagged, hey, something's going on. It looks like this may be dominant because if the gene is present, then the individual has the disease. So with this, if we say it's dominant, let's say this individual is little a, and this one's big A, little a, we're passing down a big A half the time. So that means these individuals who get the big A are automatically affected. So this is going to be a heterozygote in response to this, though. So it is heterozygote. But to change this up, say if this disease was a recessive, if there's a little a, and this individual is big A something, in order for these offspring to get this disease, it need to have a little a here. But the offspring can get either, if we use a Punnett square, big A, little a, little a, little a. We get big A, little a, little a, little a. Big A, little a, little a, little a. So 50% can get the disease, but we get a bunch of carriers. And we have no carriers in this. So this is not right. So that's why in recessive genes, recessive disorders, we would get these carriers and we would see some of these individuals as a carrier, but we don't see that. So the disease has to be dominant. If we cross this would be the same exact cross. Little a, big A, little a. Big A, little a, big A, little a, little a, little a, little a, little a. 50% are A, and have the disease. So at this, and we get no carriers. So that's why we get multiple individuals with the disease, but no carriers. Okay. So that is it for today. So for this chapter, make sure you know the different types of inheritance: dominant, recessive, incomplete dominance, codominance, epistasis, and so forth. And make sure you know how to use a Punnett square. The weekly activity and the discussion will help you with this.